Welcome, ladies and gentlemen, to the what number are we on? About 104 or so.
That's about right.
Northern Virginia Soil and Water Conservation District Green Breakfast. Green breakfasts have been going on, as I said, for a long time. This is an idea of one of our previous directors, and it has been a huge success. Ordinarily, we meet in a restaurant called Brian's Grill, which is uh, right opposite the main entrance of George Mason University. But because of the situation that we're living in these days, the best we can do is do it on a computer. What you're missing is breakfast, and the the young people who are going to uh, talk to us this morning. That's too bad. That's a free meal from them. We would have to pay. So in any event, welcome to the group. Um, I don't know how many people are here. I count the little squares, I guess. That'll tell me. And uh, I don't know.
About 32, Harry. I'm sorry to interrupt.
32. Oh yeah, okay. That's pretty good. That's not bad. We've had worse. Um, so I am stammering and stuttering. I don't know what else to say, so I'm not going to say anything. It's up to Ashley and Laura.
Okay, well, thank you, Harry. And good morning to everyone. Our numbers, we do still have people that are getting their last cup of coffee, I'm sure, and are planning on joining us as well. We are going to turn it over to Ashley, who is going to introduce our young environmental leaders and some of the programs and projects that they've been working on over the last several months. And before then, we did want to invite anybody who has a burning announcement or who may not be able to stay until the end. We are inviting you to share any announcement that you may have in the chat. And we'll take a few minutes to just get those in. If you have anything that you would like to share shortly here. Otherwise, we will be taking time for announcements and questions at the end of today's program as well. So with that, I will be monitoring the chat today. So if you have any questions that you would like to ask, or if you have any technical difficulties, please feel free to send a note to the host in the chat and you will reach me. So with that, I am going to turn it over to Ashley. We're very excited that everyone is here this morning and that you're helping. We look forward to this wonderful presentation. So thank you.
Again, everyone, thank you so much for joining us this morning. My name is Ashley Palmer, and I'm the district's conservation education specialist. And I have had the unique opportunity over the last year to work with all four of our presenters on different projects, and they have continued to amaze me in what they think about the environment and the progress that they've made and the new things that they're willing to try. And when I asked all of them to present today and they all said yes, I was so excited that you would also get the opportunity to see all the things that they had accomplished. As we're moving between a couple of different speakers and a couple of different presentations, I appreciate your understanding as we go through perhaps some technical difficulties or some slowdown as we switch some things around. We are talking primarily about two different programs today, so I'm actually going to go ahead and start sharing a presentation, and I'll talk to you a little bit more about the first one. Alrighty. So I hope everyone can see this just fine. The first program that we engaged with these students in the last year is called the Youth Conservation Leadership Institute, or as we refer to it, YCLI. And that spanned the school year from 2019 to 2020. And we had two students join us for that, Carolyn, Rohr, and Nimesh, Rudra. And for that time, we worked with the State Association, the Virginia Association of Soil and Water Conservation Districts, to create a program in which these students spend at least 20 hours working on community service projects and developing their own project that they wanted to see to fruition. So I'll start with our first presenter today, who's going to be Carolyn. And Carolyn has had some difficulties with her computer going a little slowly, so we'll be patient with her, but I'll also go ahead and be her slide switcher today. So she's going to let us know when it is time to switch around. Just to introduce Carolyn and her amazing project. Carolyn is a senior this year, and she has had some incredible experiences. She has a lot of activities in sports. She's been very active in the environment, and in the past year that I've known her, I've seen incredible growth in her, in that she now understands better what she wants to do when she goes to college next year. She's already got some applications in, very exciting, and her time with us has been really incredible. So Carolyn, I'm going to hush now, and I'm going to pass it over to you so you can talk to us all about your project. And then just whenever you're ready to switch slides, you just let me know.
Hi, I'm Carolyn Rohr. I'm a senior at Centerville High School in Fairfax County. And for my Youth Conservation Leadership Institute project, I decided to create an education plan for middle school students on the dangers of microplastics in the Chesapeake Bay. Next slide. So what was my project? My project was to develop and publish a lesson plan and presentation for middle school teachers to use in the classroom to educate their students about the dangers of microplastic in the Chesapeake Bay, because they can reduce their own personal plastic impacts. Originally, I was hoping to create like some sort of a club or something, but I had a very unclear plan going into it because I wasn't quite sure what exactly I was looking for my project, and Ms. Palmer really helped me narrow it down and figure out the best way to approach creating like a lesson plan, and just she really helped me refine what my project idea would be. Next slide. So how does it work? So like I said, I partnered with the Fairfax County Soil and Water Conservation to create a lesson plan. And so the presentation is located on their website for any teacher to have access to. It includes like the instructions for activities along with the presentation. Originally, I had wanted to be there in the classroom presenting, but because I had my own school I had to attend to, it would have been too difficult for me to leave and go present. So I ended up creating a way for teachers to be able to do the lesson on their own with their own materials. Next slide. This is more on how it works. Like I said, my original plan was to go to the classrooms. Next slide. How my plan changed. Yeah, originally I was thinking I would create like a club because what my inspiration was I attended several summer camps from the racking my brain, Chesapeake Bay Field Station. I attended their summer camp, so I spent a week learning about marine science, and I thought it was an incredibly interesting experience, and I took a lot away from it. I learned a lot from getting like hands-on work and doing fun activities, and I felt like I wanted to bring that to a bunch of like my peers and other fellow students in the Fairfax County area and just in my community, because I wanted to offer a chance to have more hands-on learning. And I thought that by creating like a club or some sort of after-school program for younger children, it would get them more involved in marine science. And like I said, I ended up going with an in-class program on educating about microplastics and the dangers of them. Next slide. Yeah, my reflection. I was sad because due to coronavirus, I wasn't able to actually see my project used in the classroom, because by the time it was finished, schools were already beginning to shut down. So I'm hoping that maybe not this year, but next year, classes start using them. The main reason I don't think it could be, well, it could be used, but there are hands-on activities that would require tools and like paper that you just have to hand out. So I'm not quite sure if it would work this year, but hopefully next year when everything is calmed down. Next slide. And this is my actual presentation. This is my actual like lesson program. So the dangers of microplastics. What are microplastics? Microplastics are very small pieces of plastic polluting the Chesapeake Bay. Microplastics are classified as pieces of plastic less than 5 millimeters long. Yeah. So where do microplastics come from? Microplastics end up in the bay through many different sources, like degradation of larger pieces of plastic, microbeads found in cosmetics, and synthetic clothing. How do microplastics end up in the Chesapeake Bay? So there are 3 main ways that microplastics will enter the bay: plastic in landfills, littering, and products that go down the drain. And so off to the side, I used a toothpaste example because I found that that was a big contributor of microplastics, because in toothpaste, some toothpaste brands include microplastic beads to help your teeth. But when you end up spitting it out to the drain, it ends up getting washed out to the ocean, and it stays in the environment instead of like breaking down. Next slide. So here are why the microplastics are bad. So microplastics, or any plastics in general, they can absorb the toxins, like toxic chemicals, and they release them into the ecosystem and into the water, which is harmful to the plants and animals in the ocean. And also, microplastics are often mistaken as food by other organisms, and they don't break down in the body. They end up building up, so they get passed along the food chain, which is what that example is off to the right. So if a group of plankton eat a bunch of plastic, and then these fish keep eating the plankton that have plastic in them, they'll slowly start building up their own plastic in their body, which will, work its way into the larger animals, and then suddenly everyone is filled with plastic. The dangers of eating plastic, if that doesn't sound scary, is it will like I said, it builds up. You can't digest it, so it ends up building up in your stomach and your gastrointestinal tract of organisms, and it'll basically trick the animals into thinking that they are full and that they don't need to eat anymore. So it ends up tricking them into thinking that they don't need to eat, so they end up starving themselves to death. And so yeah, microplastic contamination doesn't help. Yeah, and then here is a short video. I don't think we need to watch it, but this was to help kids who are more visual learners, like if they want to listen to a video, maybe watch something that shows it firsthand. I thought that this would help stimulate them and get them interested, but we don't have to watch the video right now. And then here was the question I want to ask the kids, what could you do to help? Microplastics are one of the biggest pollutions in Chesapeake Bay, our watershed. So following this, I'm going to talk about ways that they could help, because I want them to think about their own impact. Oh, we want to watch it? I guess we can go back and watch it. Myself during the video. I'm not sure if the link is working. I don't know why. Could be linked to the picture.
Could you hear the video?
No.
Okay, it was very inspirational. We'll try and go ahead and link it in the chat so that everybody else can see it as well. I think with our WebEx platform, it doesn't allow us to share the audio like that.
Mm-hmm.
So I will just go back to your presentation.
Okay. Yeah, I didn't switch to the video. It was still on the slide for me, at least.
Yeah, that was very interesting. I'm glad that we gave that a shot, and I'm glad that others were interested in seeing it. Actually, it won't allow me to share again, so I have to restart our sharing. I appreciate your patience.
We're waiting, Carolyn. This is Laura Grape. Can you tell me what the name of that video was? And I will look for it and try to include it in the chat so folks can take a look at it here in a little bit.
I don't remember what it's called. It's from National Geographic, and I'm pretty sure it is just titled Microplastics or something. Let me try and find the video.
Okay, I'll look for it, and I'll see if I can find exactly this one that's 2 minutes and 52 seconds long. That's a helpful indicator. So I don't want to interrupt you from moving forward with your presentation. Thanks, Carolyn.
Thank you. Okay. Let me decide. Yeah. Okay. So here are just some ways that I wanted the students to think about how they could help. So reduce your use of single-use plastics, recycle properly, avoid products like toothpaste that contain microbeads, volunteer in river and stream cleanups, and most importantly, spread the word of microplastic injuries. And so yeah, so I didn't want to just tell them about the dangers. I wanted to tell them how they could actually help. I want them to take away what kind of an impact they could have on the situation. Next slide. And then I included this in here because I wanted the teacher to talk to the students and ask them what their biggest takeaway was, just discuss like their thoughts on microplastic. Because I wanted this to kind of be like almost like a little test to see were the kids actually paying attention? Are they actually taking anything away from it? Like I wanted some engagement rather than them just sitting back and watching like a presentation without ever actually, thinking it through. So I included the discussion there. Next slide. Yep. And then these were the two activities that I had. So one is a Kahoot that is just a fun game that a lot of us play in school. It's a nice like quiz type game that has a bit of like a competitive aspect to it. And I found like a lot of people, at least in my classes, have been very active when it comes to Kahoots. So I thought it'd be a fun way, like I said, to like test whether they're paying attention or not and just kind of go over the information that was covered in the presentation. And then pom-pom making was just a fun hands-on activity for the kids that was supposed to represent like I have it linked in the final slide where the artwork came from. But yeah, the pom-poms are an activity from National Geographic again. That's supposed to like replace single-use plastic balloons as party decorations. So just tying into the single-use plastics. And that's it. So thank you. And then in the following slides, I have all my links and the credits for the slides.
Thank you, Carolyn. Yes, she actually does have all of her resources in her slides, and we actually have her entire presentation available on our website. It seems that Laura has already linked that in the chat. So you can see her entire presentation. You're free to use it. She's created it for everyone to use so they can get their hands on it. And I'd like to say that one thing that Carolyn doesn't talk about enough, which is a huge credit to her, is the fact that she actually made this in accordance with the Virginia SOLs, which as an educator I can say is incredibly challenging. She came to me and she had this great idea that she wanted to jump in with, and we of course had to talk a little bit about if we want schools to use it, what we kind of have to shape it to look like. And she did her research for what those SOLs are and what she wanted to meet and what her goals were. And that part of the process, which she doesn't often include in her presentation, took so much work from her and just showed so much drive that she thought that something was so important that she put all this extra effort into making sure that it was something that would be actively used in classrooms. Our next speaker is going to be Nimesh, but before I get him all set up, I think we have some time if we have any questions for Carolyn. If we have a couple, we can ask those, and we can also do more at the end because we have a couple presentations today.
We had just a couple of questions, and it looks as though Carolyn has done a great job of keeping multitasking, which is better than what I could do for certain. And she may have already answered some of these, but for the benefit of those who are not necessarily watching the chat box, there was a question about where the artwork came from, and Carolyn did share on her last slide where those images did originate from as well. And there's one additional question related to your slides, Carolyn, that just popped up, which you may have seen, which is what were the slides made with? I think they're talking specifically about what program you used, whether it was Sway or Slack or not Slack, but PowerPoint or Hoodly Doodly. I don't know. There's all sorts of new names out there these days. Would you like to answer which slide, what software program you use to create your slides?
Yeah. Oh my God, what just happened with this? Yeah. I used Google Slides to make the presentation, but I downloaded a template from a website online.
Okay, excellent. Well, thank you very much. Also, there is a question related to, are you hoping to coordinate with any of the middle school teachers this year?
Unfortunately, no. I don't really have any contacts. Like, I don't have any way to really contact them, but it is up there on the slide. If I had a way to contact them, I definitely would.
Okay.
I'm looking at a couple different colleges right now. I would like to major in both film and marine science. So I'm looking at schools that offer both programs. Right now, I think my biggest one is the University of Delaware.
Okay.
Yes, the Kahoot is available, but it's connected to the slide, so you'd have to find it through the presentation.
And Kahoot, I have to tell you, I myself am a mother of middle school students. Kahoot is the way to go. It is an awesome program, and with all of the these days. If you were to ask which one is my kid's favorite, it is absolutely Kahoot. Carolyn, there was one other question that was asked, which you did answer in the chat, but what are the pom-poms made of?
I remember quickly they're made out of tissue paper and pipe cleaners. It's actually interesting because that finding that activity was one of the hardest things for me because one of the things that I had to follow was that I think it was like the No Child Behind. I had to find an activity that schools with like a lower budget could also do because originally I wanted to like include a way to make paper straws. However, the material some schools probably wouldn't be able to get their hands on because they needed like paraffin wax. And so it took me a while to find an activity that both connected to what I was talking about, but could actually be done in every single classroom. Because if I had to have it so that if one class could do it, all classes could do it.
I really appreciate that you took that inclusion and equity approach, Carolyn. That is wise beyond your years for sure. There was one additional question, and this may be something that would be great for everyone to kind of pitch in on and through the chat is there was a question about there's a statement made that a source of microplastics is in synthetic clothing, such as fleece. And is there any recommendations or alternatives that you could switch to avoid shedding microplastics each time we wash our jackets? Carolyn, I don't know if you have any ideas, but otherwise, if not, I would like to encourage that those who may have some suggestions, chat that up in the chat box. And if there's interest in breaking out later to talk about that a little bit more, we could potentially do that after today's program. Any ideas, Carolyn, through your research?
So, it was really hard to kind of find. I found like a bunch of stuff talking about this synthetic clothing. I'm not entirely sure if it's from washing it or just from like throwing it away. So I'm not quite sure. I mean, I guess a good way to avoid if you're concerned about that. I'm not quite sure if it is from washing it or just from like throwing it away. I guess just like look for more organic material. Like if you want to wear a cotton jacket or something, I guess just maybe switch to something more like that. I don't really have a good answer to that. I'm sorry. I wish I had found that in my research.
No, please don't
I know I'm
apologize, Carolyn. Please don't apologize. You've done a fantastic job. And I think this is, again, one of those things, just like you said, there may be some alternatives that folks may want to take into consideration. So please do. I will encourage folks to continue that conversation in the chat if you would like, but I think it's a nice suggestion to know that there are some other options as well in terms of the fibers that folks might want to use. So with that, I think we've wrapped up the questions. There's a lot of accolades and a lot of kudos to you, Carolyn, on the chat. And I hope you'll take a little bit of time to look at that. Oh, I'm sorry. I did see one more question. How do you know if your toothpaste or other products have microplastics?
That is a good question. So actually on the labels, they will tell you whether or not they have microplastics in them. And that has become a little bit less of a concern because in recent times they have most products have now switched from using microplastics to using like degradable beads. So in face washes and toothpaste, like I said, it used to have it. Some still do, but you will find on the label that they do. You have to read the description and like read what is in the product. And most of the times, if a product you see has little beads in them, there will be like a stamp typically saying that it's not microplastics. So just like read the packaging and read the labels and it's pretty easy to find whether or not they have it in products. But yeah, just make sure that you check in. Is there names look on the label? I'm trying to remember because I had that included, but I had to cut it out through time on my presentation. I don't know if there's a specific name, but I feel like there is like a specific label. I'm pretty sure it is just referred to as microbeads or microplastics.
Okay. All right. Excellent. Well, I'm sure everybody is scrambling upstairs to go look for their toothpaste and see what they've got on their label there. With that, I'm going to thank you very much, Carolyn. I'm going to turn it back over to Ashley to introduce our next speaker.
Thank you. As one last accolade for Carolyn, I did want to say that when the pandemic hit everybody on that second week of March, her program on microplastics was actually the only virtual program that we had available on our website at the time. So it was a really great resource to kind of point people to as they were still looking for something to learn as things started to get a little crazy. So that was definitely a huge kudos to Carolyn. Switching gears a little bit, but still kind of staying in water, we get to talk about our next presentation from Nimesh. So Nimesh, I'm going to make you our presenter, so you can go ahead and get your slides ready while I introduce you. So that should be switching over now.
Yeah, I got it. Thank you.
Okay, perfect. All right, so for as a brief introduction, this is Nimesh Rudra. He is a sophomore at Thomas Jefferson High School for Science and Technology. He's interested in pursuing a career in the STEM field, and his hobbies include hiking, playing basketball, reading, running, and he's part of the TJ cross-country team and has hiked many trails across the United States. He's a big fan of the Washington football team, and he volunteers at the local county park. He also has the unique perspective of joining us both for YCLI and for independent research projects we'll talk about later. So his perspective is particularly valued. With that, Nimesh, I'll hand it over to you so you can talk about your project.
Thank you. So I'm Nimesh, and my YCLI project, I looked at water quality in the Accotink Creek, which is a water body that flows through Northern Virginia, and our ecosystem's health is dependent on it. So the objectives of my project were to test the Accotink Creek based on 4 chemical tests, which I'll get into later. And then I cleaned up the Accotink Creek, and then I tested the water for the same qualities again, and saw if there was a difference between the 2 tests. So basically, what I was looking at was to see, does the pollution around the creek affect the actual health of the creek? So my outline was that I tested in 4 different locations along the creek before the cleanup test. They were pH, dissolved oxygen, phosphorus, and nitrogen levels. I organized a cleanup and collected trash with some volunteers. I then went to the same locations again and tested the water, and then I looked at changes. This is done with Northern Virginia Soil and Water Conservation District and Respond. So for the before test, I got the equipment from my high school, Thomas Jefferson High School, and I partnered with the Fairfax County Park Authority to get permission to test their waters, and this was done on February 1st. So the results are in this table. Most of the tests came healthy, except phosphorus slightly high, and mineral oxygen was higher than usual. These are a couple of pictures of me testing and tapping the water. So for the cleanup, I got 5 student volunteers and 2 adult volunteers. I got the cleanup supplies from the Fairfax Clean Water Council, which basically just gives people who want to organize the cleanup supplies. And they were mainly like safety gloves and trash bags. And we got 7 bags of trash. They're mainly plastic, but we even got like tires and other large pieces. And then on February 22nd, I went into the same locations again and tested. On the next slide is the comparison. So most of the tests remained the same, but dissolved oxygen went into a more healthy range. The pH went from slightly basic to slightly acidic, so slightly over 7. I'm sorry, I meant slightly acidic to slightly basic. And then the nitrogen phosphorus remained the same. So overall, I helped the community by picking up the trash and helped the stream get better after the cleanup. I also learned more about our environment and help our ecosystem in Northern Virginia, and I hope to do conservation projects in the future. Thank you.
Okay, we do have time for a couple of questions for Nimesh regarding his project. If anyone has any questions?
I actually have a question for you while we're waiting for some more to come in. And I just say this because I know I worked with you and I know what the answer is probably going to be. But what was the most challenging part of your project?
Okay, so for the project, I had to talk with a lot of agencies and organizations like the Fairfax Park Authority and Fairfax Clean Water Council, things like that, to get permission for the test, to get equipment for the test. And it was a lot of emailing back and forth and a lot of talking with them to make sure that I could actually do the project. So in the beginning, I thought it would just be a quick, straightforward thing, but it took a lot more time than.
Nimesh, we did have a couple of questions that did come in. One being, would you have expected after a large storm that the results would have been different?
They would probably be different because after a storm, there'll be more runoff. So like fertilizers and other pollutants will get into the creek. But when I tested, there was no the weather conditions were about the same.
Okay. And did you feel as though that that the cleanup was the sole influence, or were there other factors like temperature or day of time, for examples, that may have influenced your results?
I went for both the before test and the after test. I went and on a Sunday. They were both in February. One was like in the beginning, one was like mid-February. There was probably other factors, but the cleanup was definitely one of the factors.
Okay, excellent. There's a lot of questions about whether or not you know about the Friends of Accotink Creek, which is a volunteer-led group in Accotink Creek, and they're focused very heavily on enhancements and to water quality, among other things, as well. Are you aware of the Friends of Accotink Creek? They're doing some of their fall cleanups right now.
Oh, I've never heard of them, but I'll look into that. Yeah.
I think you would. I think you all would have a lot in common. There are a lot of questions that are coming in, and we will have time for some additional insights here toward the end of our program as well. Let me find one more here. There was a question, actually, if we could go back to the slide that showed the results from the before. Looks like someone may want to take a good look at that question.
Sure.
And then also, what kind of follow-up experiments do you think you might want to do with this information?
I wanted to probably, I would, if I could, I would probably do cleanups to see, because I only did one cleanup, so I was hoping to do like 2 or 3. I didn't just have enough time to do that, to organize and get all the timings right for all the volunteers. But if I was to do it again, I would organize more cleanups to actually see if there's a real effect of the cleanup.
Okay.
The table
Excellent.
for the differences.
Excellent. And did this kind of follow through on your hypothesis? Were the results kind of what you were expecting, what you hypothesized, or did they come out as a surprise?
I did hypothesize that the levels, the water would get better, which it did. But for most of them, they remained the same. But for dissolved oxygen, it did get better. So I guess I kind of went through what I.
Okay. Well, thank you very much, Nimesh. I'm going to turn it back over to Ashley.
Thank you.
Thank you again for sharing your project with us. If anyone wanted to see his presentation again, that is also on our YCLI webpage, so you can also view that there, as well as some fun pictures of his cleanup. For YCLI, I did just want everybody to keep in perspective that the students working on those projects had very short windows of time that they could be active, and then also the hours commitment was about 20 hours. So because Nimesh was actually traveling to and from sites and coordinating large numbers of people, even though he only went to his site physically 3 times, it did take a large number of hours to do that. So when he talks about wanting to go back again and get some more data, we know that's very time intensive, but of course we'd all like to see it in the future. I'd like to switch gears a little bit and talk about another program that the district did this past year. This summer, we saw a need in our high school students to still be engaged with the district, to still be learning, although making things virtual had really changed everything for everyone. And we decided that we were going to do some research projects with high school students and give them the opportunity to really research what was important to them. So we decided to do that this summer in a 10-week program called the Independent Research Projects Program. It's not a very catchy name. If you have any recommendations, let us know. We'll keep it in mind for next year. But we extended the offer to a lot of high school students who had worked with us in the past. So Nimesh had actually been in YCLI. Cynthia was supposed to be doing an internship with us before that was canceled. And Becca had done a very large storm drain labeling program. So they all ended up joining us for this. This 10-week program was very time intensive. intensive. It lasted the entire summer. We met once a week, every week, to talk about our projects, get input from our cohort, get advice on what we could be doing for our projects, and sharing our results along the way. So that way, if there were any hiccups, we could catch those early. And then we spent a lot of time poring over our data and spending some time thinking, what do our results really mean? What kind of changes does this actually indicate? So the next series of projects that you're going to see are very highly research intensive and involve those 3 students who'd worked very hard. As I introduce her, I'm going to go ahead and make Cynthia the presenter so that she can go ahead and get everything ready. While she's pulling that up, I'd like to introduce Cynthia Ma. She's a junior at McLean High School with an interest in environmental conservation and climate advocacy. She's currently a director of the Youth Climate Action Team and a volunteer at the United States Department of Agriculture. She also recently started an environmental blog to write about environmental concerns and some of her more memorable experiences. Aside from her environmental interests, Cynthia also loves to participate in outdoor activities with her family and friends. Without further ado, I'll let her tell you a little bit more about her project.
Okay, can you guys all see the screen? Okay, so my project was called the Efficacy of Different Erosion Prevention Methods. I'm going to give you like a quick introduction about the environmental impacts of erosion and kind of why I decided to research a lot on this. So basically, accelerated erosion can be caused by construction sites of roads and buildings and recurrent walking in a certain path. Normally, undisturbed soil will have a natural ground cover that protects it, but after time that ground cover can deteriorate due to accelerated erosion and human activities. The impacts of erosion are that it can cause the destruction of natural habitats, the removal of vegetation, and the transport of eroded soil into bodies of water. So given these impacts of erosion, I knew that it was extremely important to understand the ways to prevent erosion and which ways may be more effective than others in a certain geographical location. So my project basically were these 4 boxes that had that measured the slope degree and also rain intensity. So those were the 2 factors I kind of focused on for my erosion project. So box 1 here is elevated at 7 degrees and also has a 1-foot rain height. Box 2 has a 7-degree slope and a 2 feet rain height. Box 3 is 12 degrees slope and a 1-foot rain height, and box 4 is 12 degrees and 2 feet rain height. The purpose of changing the slope degrees and the height of the rain was basically just to measure which erosion prevention method will work best under the conditions of like a higher slope or a lower slope, and also which one would work better in a greater or lower rain intensity. So to measure rain intensity and how I measured how the rain intensity would be for each of these boxes, in the picture on the right here, I kind of drew a diagram of how I watered each box and how the slope and the rain height kind of affected each box. So after several days, so like over like every other day, I would measure the soil and to kind of give an estimate of that slope change. So I did this by using a thin rod that I stuck into 2 sides of the top part of the box and also 2 sides at the bottom. So then I'd measure how much of the soil reached up to like a part of the rod, and then I would subtract those measurements to kind of see this degree of erosion. I did it twice just to measure the accuracy of it, and that difference of depth would be the estimated measure of the degree of soil erosion. So here's some of the results. I not only got results from the data, but also from the visual observations I made on these 4 boxes. So the creeping jenny overall had a good erosion prevention methods, mainly for low slope conditions. And as you can tell here in boxes 3 and boxes 4, where there's a 12-degree slope height, the creeping jenny did not grow as well compared to boxes 1 and 2. And my estimate to why this is because due to these different conditions of slope height, it was harder for the creeping jenny to grow in that higher degree of slope. And they almost look like I just planted them on the first week. But for boxes 1 and 2, after the 9-week period, they were able to grow much larger and almost extend out of their section. For the gravel, it was also a good erosion prevention method for low slopes, but particularly for slopes with less rain. If you can tell here in boxes 4 and 3 with the 2 feet rain height, the erosion of that gravel section is pretty obvious. As you can tell, most of the gravel rocks are kind of near the end of the box, while at the top, like this is the top and this is the end, there's a lot less gravel here compared to down here. Rohr was a good erosion prevention method for higher slopes, but not for slopes with intense rain. As you can tell here in boxes 4 and boxes 2, you can barely, this is the section where the straw is, you can barely tell it's there because after this 9-week period of watering every day, the straw really condensed together and kind of contracted into this middle of the section, and it left the edges of that section really exposed. But in boxes 3 and 1, where there was only a 1-foot rain height, it was able to hold the soil a lot better. And lastly, mulch was a good erosion prevention method for both high slopes and more intense rain. What I noticed visually was that in the sections with the mulch, there was barely any shifting in the soil, and the mulch also stayed, didn't shift at all. So there was no visual observations that there was obvious shifting in that section. So here I kind of talk about how you can apply these different erosion prevention methods into real-life scenarios. So the creeping jenny, firstly, it's ineffective as a ground cover in construction sites. It would be much better for natural sites where the land has a level to moderate slope and it's not usually disturbed, to also promote plant growth. So from the results, definitely that the creeping jenny plants don't work well in higher slopes. So if you want to use a creeping jenny ground cover as an erosion prevention method, they should be in natural sites, and they're more of a permanent type of erosion prevention method compared to the other ones I'll talk about. So gravel, on the other hand, works best in areas with pedestrian-induced erosion and lower slopes. Here on this picture, as you can tell, it is next to level roads and trails. They can also be seen in various sizes near storm drains to prevent water erosion, and the larger rocks can also hold and steeper slopes. So gravel does not work in places where there are higher slopes. It can be more of a permanent type of erosion prevention method, but over time, if a lot of the gravel shifts, it may need to put new gravel in that location. Third, straw is a great temporary ground cover due to its inability to trap sediments on a soil slope, due to its ability to trap sediments. But however, in a long period of time with heavy rain, the straw will degrade and leave some parts of the soil exposed. So it is often used temporarily. In real life scenarios, you often see straw used in construction sites or along roads because it is better as a temporary ground cover. And lastly, mulch, unlike the other 3, is capable of withstanding steeper slopes, and it would work well in both hilly landscapes and more along walking trails. But obviously, like mulch and straw, they likely need to be, you can't reuse them for a super long time, and they will need to be recovered on that area. So the conclusion of my project is that no matter what location you are looking to use a erosion prevention method on, it's really important to understand the qualities of that erosion site, to understand which soil, like the soil type and the climate of that site location, to know which erosion prevention method would work best for that site and it will prevent erosion most effectively. And yeah, that's the end of my presentation.
Okay, Cynthia, that was a great job. I really appreciate the your ability to draw the conclusions and to create a fantastic project in order to collect data and certainly analyze it and come up with some different different ideas of erosion control and identifying what based upon your experiment, what worked well and what didn't. So there's a couple of questions that did come up. One is, what type of mulch did you use or measure?
I think I just use like, that's a good question. I'm not exactly sure the type, but I think it's like the most typical one that's often used in like gardening. And yeah, I'll have to check that because I'm not exactly sure.
Let me ask Cynthia, were they kind of like blocks or were they more shredded?
Oh yeah, no, they were more shredded, yeah.
More shredded. Okay, excellent. There was another question in regards to the use of Excel. Can you talk a little bit about what you did in terms of identifying trend lines? There was a question about whether you used Excel to calculate the 4-point linear regression erosion trend line.
Yeah, so over, because I measured the measurements of that soil every day, I kind of put those, the height of the soil into a spreadsheet. And then after this 9-week period, I took like the final and initial measurements, and I used that data to create the trend lines I used for the graph that I made.
Okay, excellent. Excellent. And then one of the questions, there's two more questions that we'll ask here. One was, how did you decide how much water to apply, and over the certain time frame? How did you come up with those two decisions? How did you come up with those two variables?
Okay, so I use the same quantity of water for every box, and basically the amount I used would make sure that it covers most of the soil, like obviously not flooding the box either, but also making sure that there is like, it will, because this 9-week period was not exactly a very long period to measure erosion, so I did have to use an ample amount to make sure that over time there was a shift that I could see visually. And the water quantity definitely would have made a huge difference, because the amount of water I used, I could see like the intensity of the water shifting some parts of the soil. And I'm guessing that if I used more water for each box, there would definitely be more of a shift, especially with the angle I was watering it. So, yeah.
Okay. Excellent. Excellent. Well, thank you very much. Yeah. The big part of a lot of these different kinds of projects is, again, working kind of through that scientific method and identifying what those independent variables are going to be in regards to what you're going to be measuring and then what you're going to be kind of using as a constant. So I think it's great to hear just what you're doing and then thinking about, if you were to replicate this, what you might do differently next time too. That's fantastic. Final question has to do with use of creeping Jenny. And if you could talk a little bit about why you particularly chose using creeping Jenny. And if you have, I think actually what I might suggest, if others have suggestions for other plants, that you include those in the chat. But Cynthia, if you have any other recommendations or features of the plants that you might recommend landowners use in landscapes to prevent erosion, that would be great. But that was a whole lot there that I just asked you, so let me back up just a little bit. Number one, why did you choose Creeping Jenny? And number two, what features of plants might you suggest landowners consider using for erosion control?
If I could just make a comment on the Creeping Jenny real quick, I guess I should probably say this for all of the independent research projects. These were all done entirely in the pandemic, and so it was best that instead of sending our students into stores to get their own supplies or things like that, that the district actually provided all of these materials. So I think that Cynthia could also talk a little bit more about Creeping Jenny. But a lot of the materials are basically what the district had on hand or what we were able to get in the short time frame that we had when we were doing our research projects, and what I could reasonably deliver. So you had me in a mask and gloves dropping supplies off at everybody's house and then picking them up at the end. So I think all of them would have chosen different supplies than I could have offered if they got the chance to redo their projects. But just as a little bit of background for why we chose some things over others.
Gotcha. So it was a given to you.
It is what they were selling at Home Depot.
Okay. Cynthia,
Cynthia can talk a bit more about that.
Yeah, Cynthia, do you have any recommendations for plants that you might suggest for use in erosion control?
So I actually did do some research on the Creeping Jenny, and it was commonly used as ground cover. I don't know any other plants that may be effective, but I'd say if I were to extend my research on this project, I'd definitely do something with different types of ground cover and measure how effective they are.
Okay, fantastic. Well, I'm going to actually put a link in the chat box here in just a little bit, because we do have some information on our website in regards to different plants that we encourage use for slope stabilization as well as for erosion control as well, kind of faster growing native plants as well. So we'll send that to you. I know Plant Nova Natives also has some information that we've worked together on as well. Cynthia, this was a great presentation, and again, a great example of the use of scientific method and coming up with conclusions based upon the results. And I think that this is a topic that is near and dear to the Soil and Water Conservation District's heart, because we get this question a lot. So thank you very much for a great presentation and a lot of great work.
Thank you.
Alrighty, for our next presentation, we get to go back to a speaker we've seen before. Nimesh, I'll go ahead and make you the presenter again. There you are. And as we pop over, when we first started working with Nimesh, he was a freshman who'd been in school for 2, maybe 3 months. And so I was absolutely amazed in how he grew in between his projects and how he intends to grow in the future. And this, I think, was a really good, out of left field, totally different than his last research project that was fun to work on with him.
Yeah. So I'm Nimesh. I presented last time, so you can hear me. But, so I look at the effects of microhabitats on insect biodiversity in Northern Virginia. So I chose this project because insects are often overlooked, and I just want to know the diversity of insects and how our actions are affecting insects. So the objectives was to study insect biodiversity across Northern Virginia, and then look at the effects of the microhabitats on the biodiversity, and then analyze how urbanization, deforestation, and how actions are impacting the environment. So I selected 6 different sites for sampling for the insects, 2 urban sites, 2 forested sites, and 2 grass fields. The sampling area was 0.8 meters and 0.8 meters to survey for all the insects in each of the 6 sites. I used the iNaturalist app to identify the insects. So what I would do is I would take a picture of the insects I found, and the iNaturalist app would give me suggestions about what species the insect could be. I picked the one that most was similar to the one I found. I then log this data once a week, and then group them into these insects into conventional classifications, so the words that we use in day-to-day language. And this was done with the Northern Virginia Soil and Water Conservation. So I sampled on Wednesday or Thursday, depending on weather conditions, because if it rained the previous day, or if it was raining on Wednesday, I didn't want to go on Wednesday because I would disrupt the data that I would collect. So I went on Thursday, so depending on the weather on that day. So I went between 6:30 and 8:30 because that's when it was in the morning. It was really cool. There's not many people outside, so the lower temperatures helped. So I can get more insects. I can get more data. So I surveyed each site for approximately 25 minutes. They were all within 2 miles of my house, and I went every week from June 28th to July 19th. And I used a hula hoop to set the boundaries of the sample, the 0.8 meter to 0.8 meter sampling area. I then, some conditions I use, instead of counting the because colonies have lots of insects in them, so instead of counting them all individually, which took a long time, I counted them as a whole entity. And the sites I used were, for the forested sites, I used a forest in a nearby park called Longley Park, and then I forest off the W&OD Trail, which is like, I think it's a 70-mile trail that runs through Northern Virginia. There also the nearby park. There was a field, my backyard, a cul-de-sac, and a giant store parking lot. Here are some pictures of some insects I found. So here on the right, you can see the earthworm. This is a woodlouse, and then here's a moth, and this is a Mesopotamian fly. It's very interesting. Moving on, here are some other pictures. You can see a spider in the top right, and then there's a dragonfly on this leaf. There's all kinds of insects I found. So for the data for the Grassfield, so as you can see in the pie chart, it was mainly white flies, which are these tiny white flies that fly around. Those are 60%, and another 26% were mosquitoes. So most of the insects found in the Grassfield areas were flying insects, which I'll get into later of why. In the forested sites, there was a lot more diversity because you can see there's more slices in each pie. There was woodlouses, there was mosquitoes, and then there were spiders, dragonflies, so there's all types of insects. And in the urban, it was predominantly mosquitoes with 73%, and then occasionally I found some ant colonies and some birds. So in conclusion, the forested sites had the greatest biodiversity, followed by Grassfield and urban. This is because the forested sites had the greatest shade cover, and because of more shade cover, there was lower temperatures, there was less foot traffic because people don't want to go in areas that have more vegetation. There was more nutrients and food sources, and there's more places to hide from predators and threats. The Grassfield had lots of flying insects because it was mainly open, so they couldn't, to evade threats, they had to fly because there was no way they could hide in any like plants. And the urban had the least biodiversity because it's like a hot tarmac. It's really high temperatures, and there's lots of people walk there and cars there all the time. So it's really not suitable for many insects. But for mosquitoes, it is because nearby urban sites, there was like these compacted yards, our yards, we compact the soil for plant growth. And when it rains, puddles form in those areas. So the mosquitoes use that as breeding grounds, and they could live in these environments. So the constructions and urbanizations in Northern Virginia is driving these insects away, and we have to do a better job to preserve these environments to make sure that these insects are safe. And here are a couple pictures. That's it. Thank you.
All right, that was a great presentation, and I'm going to echo something that was just said, that this is a very revealing result around the count of mosquitoes in urban and suburban settings. I think you're spot on with a lot of that, and also some of the human-created sources of water and habitat, perhaps for mosquitoes. I know for myself, for example, I have to go around and turn over things pretty periodically, especially with all the rain that we've had more recently, in order to try to get rid of the suitable habitat that I'm creating for mosquitoes as well. So couldn't figure out for a little while why I was getting so many mosquito bites in one particular area, but I certainly can understand where they came from once I found that pot of water. Harry, I see you've got something that you would like to say. Go ahead.
Yes, if I might. I'm particularly interested in this one because I hold very strongly the belief that insects are the most important animals on Earth. They are certainly more important than human beings, which are the most destructive animals on Earth. But insects pollinate all of our food. Not all of it, but a great deal. If we did not have insects, we would eventually all starve. Given the uselessness of human beings, maybe that's not a bad idea. But nevertheless, insects are terribly important, and a further study of insects and their habitats and well, very good.
Harry, thank you. It sounds that we've got a little bit of feedback coming in, but I think we got your point in terms of in reinforcing the importance of insects for our human survival, but also just in terms of our ecosystem survival and biodiversity as well. Nimesh, we've got a couple of questions that have also come in. Do you have any thoughts on why you might have had a biodiversity spike in week 2?
Oh yeah, that's because in the first week I wasn't really looking. I wasn't actually like digging into the soil and looking at under the leaves and under the soil to look for insects. But then in the second week and the third week and the fourth week, I was looking more into it. I started going earlier in the morning. Like before, I'd go like at 8 o'clock in the morning, but then I started going like at 6:30 because there was a lot more insects there because it was so much more cooler.
That's what they say, don't they? The early bird gets the worm. I guess that's when the insects are pretty active as well. And then there's another question in regards to, I guess, more of your urban setting. Were those grassy areas mowed fields, or were they more meadow grasses?
I think they were mowed. Yeah, I think they're periodically mowed.
They were more like lawn landscapes. Okay, excellent. And then one final question before we move on here. Do you have any, there was an interest in sharing your method that so other students could replicate it. I think Kim, the teacher over at the public schools, may be reaching out to you for some additional information on your methods and strategies, and if this is something that would be replicable. I certainly think that that's the case, if you would ask me.
Yeah, definitely. I can share my methods.
Okay, excellent. Well, thank you very much. I will turn it back over to Ashley and Nimesh. Great job. All of these presentations, all of these projects have been really insightful, and it makes me very excited for the future of our conservation field.
Thank you. What was really exciting, I think, about the independent research projects is that since everyone saw each other every week, we were constantly able to revise our projects and change things up. And I should have that opportunity when he said, we didn't find very many bugs. And we asked, well, are we looking under leaves and things like that? And he's like, oh, that's a great idea. And then that totally changed his project. He found so many more things. So I think everybody had a moment like that where someone else gave him or her advice about their project, and they were able to put that in and make it better. So not only did they all do incredible projects, but they all helped each other to do incredible projects. Our last speaker today, certainly not least, I always say last but not least, is Becca Jeffries. She's a sophomore at Thomas Jefferson High School for Science and Technology, and she has a lifelong passion for conservation and environmental science. She's been involved in several environmental initiatives, such as being a member of TJ's Envirothon team this past year, and hopefully this next year, and being a current officer for school's Environmental Impact Club, and most recently interning at the Plastic Tree, a DC-based reuse company. Now, because we've had some technical difficulties when she presents, I believe that I'll also be presenting for her. So I think I have to make myself the presenter. My apologies. All right, Becca, this should be pulled up for you pretty shortly, and then you can tell us about your project, and then just let me know whenever you'd like me to advance slides.
Okay. So hi everyone. I hope everyone's doing well. My name is Becca, and today I'm here to talk about riparian and natural stream buffers, and to present my findings from my research project. So for anyone unfamiliar with the term, riparian buffers, or stretches of vegetation planted along a stream, are a widely used pollution prevention strategy. Buffers keep pollution from reaching waterways because when it rains and pollutants run off paved urban areas, they're soaked up by the trees and other plants before they can reach and pollute the stream. Forested stream banks are a natural buffer and often the best case scenario, but riparian buffers can be very effective if the vegetation that once existed has already been cleared for development. Okay, you can go to the next slide. So at the beginning of the summer, I found myself wondering exactly how important these buffers are and how much of a difference it creates in water quality. Coburn's Stream Valley Park backs up against my neighborhood, and I've been biking there pretty much my entire life. And sadly, there's a notable difference, even visually, between the quality of different parts of the stream. In some places, the water is completely clear, and you can see an abundance of plant and animal life on the bottom. However, in other places, the surface of the water is completely covered with algae, devoid of other life, and foul smelling. Could this be because of the level of vegetation on the stream banks? I decided I wanted to find out. You can go to the next slide. So, for this investigation, I studied 3 different sites with differing levels of urbanization, and at each site, I completed a physical assessment, which is a means of calculating stream health by simply viewing the stream and looking for telltale red flags such as excessive algae, or also good signs like shade cover, which is really important. And additionally, I measured temperature and dissolved oxygen levels. However, as I'll explain later, these are not always the most accurate tests. And most importantly, I collected and identified benthic or bottom-dwelling macroinvertebrates, and with the aid of the Alp Creek critters, was able to pinpoint the water quality on a range of poor to fair. And I collected data once a week for 3 consecutive weeks. Next slide, please. My computer might be lagging. All right, is the slide on the project description or the next one?
The slide is now on the site description.
Okay, yeah, sorry. I don't know what's going on. So the first site I considered the most urban or least natural, as one stream bank was mostly devoid of vegetation and straight into the water. Whereas the second I labeled as the pedestrian site, as it had a bridge for walkers and runners and bikers to get across the stream. And the last was the most natural by far. It was pretty deep into the woods and only had a small, barely used gravel path running near it. You can the next slide. So as previously mentioned, Site 1 was the least natural site. And as you can see in the picture, there's barely any vegetation growing on the side of the stream, and the rocks in the foreground of the photo are where the retention pond drain lets out. However, there was vegetation on the left bank. And the water itself did not appear clean even at a glance. It often had an oily sheen on top and was always very muddy. The stream bottom did not have any visible vegetation as well. It was just as bare as the bank and therefore not very firm. My feet sank pretty deep into the mud. There was a musky odor by the water and a lot of algae. And there was a moderate amount of shade, but unfortunately a moderate amount of erosion on both sides. Next slide. So, Site 2, the pedestrian site, had wide buffers and was relatively natural except for this stream crossing where the bridge runs over the water, and there's also a cement path that runs a few inches deep under the surface of the water. The water appeared brownish but not super muddy, and the stream bottom was almost completely covered with rocks and vegetation. There was also a musky odor here as well, a moderate amount of shade, and only a little erosion. Next slide, please. So Site 3 was the natural site, and it's not near any paved paths. The only development near it is a small gravel path I used to access the site. The water was brownish, and the stream bottom was extremely rocky and vegetated. There was no odor and very little erosion, and there's also excellent shade cover and a lot of trees, as you can see from the photo. And the stream was mostly shallow here, only about 10 inches deep, where I collected my data. Next slide, please. So in the end, I ended up finding no significant trends in the temperature and dissolved oxygen levels. When it came to these two tests, no site appeared to be any more or less impaired than the others. These results were not actually very surprising because both of these tests can be very unreliable. The reason for this is that both of these tests use a single water sample, and because streams are moving bodies of water, the water is constantly changing. If you take a single water sample, then the water you'd sampled is gone in a few seconds with new water rushing in from upstream. This means that they do not very accurately represent the long-term health of the stream, as they tend to only represent the quality of the water running through that site at that exact moment. Additionally, the equipment I used could have been up to multiple years old, which may have also affected the accuracy of my results. However, shown here on this slide is the range of stream health given by the Creek Critters app, which provides a lot of information about the water quality and level of impairment at each site. Since I collected data at 3 different times, I did calculate 3 different scores for each site, but by printing them out on a sheet of paper and measuring the distance from the left edge, I was able to find a quantitative value that I then averaged with the others and remapped onto this range. So as apparent, the natural site is clearly much less impaired than the other two. Next slide. So this graph shows the different organisms I found and the biodiversity of each site. As you can tell, I found a large amount of different organisms and therefore a wide biodiversity at Site 3. Sites 1 and 2 had similar biodiversities, but Site 1 had 2 more different types of macroinvertebrates than Site 2, which were riffle beetle larvae and mosquito larvae. There were a number of organisms I found at all 3 sites, as you can see, such as the small minnow mayfly, which was really common. I found those present in abundance at each site, whereas other organisms I only found unique to each site, such as the casemaker caddisfly, which I only found at Site 3. Next slide. So what does all this mean and what can the data tell us? So the natural site was definitely much healthier than the other two, but what could be the reasons for this? Well, for one, the level of pedestrian traffic might have been a factor. A lot of people go over the bridge at Site 2, and a lot of people like to fish or just hang out at Site 1, whereas I doubt many people cross through the woods to get to Site 3 very often. Since Site 3 was also relatively removed from paved paths, this could have protected it as well, as the runoff from the paths would be unlikely to reach the stream bank there. The amount of shade is also a critical factor because the colder the temperature, the more dissolved oxygen there is, so shady spots generally tend to have more dissolved oxygen than sunny spots. The water temperatures I recorded seem to depend on the amount of sun and time of day, and the dissolved oxygen levels, the dissolved oxygen test that I did can also be disregarded as I mentioned, there were no evident trends that could prove anything about any particular site. However, the stream score definitely gave a lot of insight into the level of impairment at each site. Next slide, please. So the main conclusions that can be drawn from my research are that riparian buffers or natural buffers are undeniably critical for water quality. And furthermore, the differences between the water quality at each site unfortunately lets us know that stream health is not uniform, even within a single stream. So it is essential to protect the entire stream, as 1 or 2 stretches of vegetation will not keep pollutants from entering the exposed parts of the stream. And at the end of the day, protecting our streams is protecting our own health, in addition to the natural world. So it is important to do everything we can to create buffers and keep developed land from reaching the stream banks. Next slide. So thank you so much for your time and attention, and I hope you enjoyed and learned something from my research.
That was great, Becca. Thank you so much. You did a lot of research, and it was you had a nice summer for being out there in the streams. That's for sure. This is definitely a program near and dear to my heart in terms of monitoring water quality, and is something that the district has played a large role in from a voluntary standpoint for a number of years. So thank you very much. Do we have any questions for Becca? Or maybe some that come in here. A lot of kudos coming your way, Becca. Hopefully you can see some of those in the chat. Do you think the location of sites, either downstream or upstream, made a difference?
Yeah, I think the location of the sites definitely could have made a difference as some sites were upstream of other ones. So, which might be another reason why the temperature and dissolved oxygen tests didn't get super accurate results, because in the stream valley, site one was the most upstream, then later on was site two, then later on was site three. So the water that I measured for the temperature and dissolved oxygen tests could have technically been measuring the water that had just rushed in from the other sites. So I think this definitely could have made a difference, the location in the stream valley.
Gotcha. That's great. I do have a question. Did you just generally, even just through an estimate, just how wide the buffer was at each one of those sites? Was it pretty extensive, or were there, you mentioned trails and things like that. Sometimes the width of the buffer can also cause some interesting results.
Yeah, so there was definitely the largest buffer at site three, which was the natural site. Um, One of the stream banks, there was no paths near the other side, so it was just straight woods. And then on the other one, there was definitely a wide buffer between the stream and then the gravel path I used to access it. Whereas for site one, there was barely any buffer where I accessed the stream. There, it was only a few meters from the paved path, and there wasn't really any vegetation growing there. However, on the other side, there was a relatively wide buffer. And then for site two, there was a moderate buffer on both sides.
Excellent. Excellent. It's pretty consistent, it sounds like, with what one might expect. One other question, and then there's a couple of other suggestions. If you're considering additional research, you may want to take a look at some of those. But did you consider any other chemical tests that might be an alternative to use other than dissolved oxygen that might help to explain some info?
Yeah, in the beginning of the project, I definitely considered doing other chemical tests, and I wasn't really thinking of doing the macroinvertebrate collection or test. I was thinking I could just do all chemical tests, but then Ms. Palmer explained the concept of how the single water sample isn't always the most reliable. And if I'm looking for the long-term water quality, using the biological test of the collecting the macroinvertebrates might be a little more effective for what I was looking for. So I definitely did consider it, but it seemed that the best option would be doing the collecting the macroinvertebrates and assessing those. And so that's what I ended up doing, as well as the other two chemical tests.
Excellent. Excellent.
We actually would have really liked to have been able to do chemical tests as well. And we were going to order some, but of course, with the pandemic, they actually wouldn't have arrived in time for Becca's project. So I did give her some supplies that we had in the office, but because the district doesn't do chemical monitoring, some of the supplies that Becca ended up working with were almost as old as she was. And so that's why she was saying that some of them were not super reliable. But it was great still to have that experience of trying those tests out. And if we were to do it again, we would order those far in advance and have them available.
Absolutely. No, I think that's great. Again, I think one of the things that I just really appreciate, and I want to recognize also Ashley Palmer, who served as the mentor for all of these students, and helped them think through a lot of the different, think through their approach to the projects, was very, very well done. I think the future, again, is very, very bright with these students as well. Ashley, I'm going to turn it back over to you, and to answer or ask some additional questions.
Thank you. Did you have any announcements that you wanted to make?
I'm sorry. Yes, let me go and get back over here to the chat. There are a few announcements that are popping up here in the chat. Let me go back here. There is for those who are interested in climate change, George Kralovic did include a link to a webinar that is taking place this afternoon from 4 to 5:30. The link is there. It's just a little bit up, excuse me, from the bottom of the chat. I will also try to put these, excuse me, these announcements in a consolidated form as well. There's also more information about solar in schools. They are looking to recruit some additional members in order to enhance the solar in schools initiative. And so you can take a look at their website. And again, I will include that here in the chat as well. There are a number of Ask the Experts video conferences being sponsored by Plant Nova Natives on how to use iNaturalist, planting a pollinator landscape, and foundation plantings with natives. Please take a look at Margaret Fisher's announcement there for dates, along with where to register for those different events. Let's see here. Yes. So there is, again, the links and everything for the Solar on Schools initiatives. Ashley, one of the things I hope that you might mention is when the next opportunity for YCLI might take place as well. So with that, those are the announcements that have come up so far. Again, please do keep an eye on the chat if you are interested in if there are additional initiatives that are taking place right now.
Thank you. YCLI is a school year-based program, so the application opens every year in September. It just closed about a week and a half ago, and as I've heard, we had, I think, about 6 or 7 students from Fairfax County apply. For perspective, there were only 9 students in total last year, so the interest in the program has definitely risen, and I'm really excited to hopefully see some of these faces again in that program. Certainly, we'll be doing our summer research projects again in this upcoming summer, and then more details will be coming out in the spring. Particularly on a closing note, I wanted to hear something from all of our student panelists today, all of our presenters. A lot of the people who join us for Green Breakfast are in an environmental group and they want to learn how to get other people involved. They may be students like you and they want to learn how to do projects like yours. Or it may be a teacher who's looking for resources, or we've got a wide audience here. But I think that everyone would be interested in hearing a little bit about maybe your advice for getting students engaged in environmental science and in projects like these. Nimesh and Becca and Cynthia and Carolyn, do any of you have any input on how to get high schoolers like yourself engaged in projects like these?
Okay, I guess I can start. Well, for me personally, I got really involved with the opportunities my school had. So I guess if you're an educator at school, providing opportunities for students to be introduced to like environmental topics is really important for fostering that interest in environmental science and conservation. So really just providing opportunities and educating students about that, I think, is really important.
I would say that it's so easy to get involved. Like, it's so easy to find things to get involved with. Like, I first found Virginia Soil and Water Conservation District by Googling environmental service hours. And then I did a storm drain labeling project, and now I'm here doing this. And so it's information on how you can get involved is so readily available. So I think just Googling or asking around, like, just get involved, and it's pretty easy to find ways to do so.
Yeah, I was gonna.
You can go.
Oh, okay. I was just gonna echo whatever they said, and I also was gonna say that, like, let the students choose what they like, because like when we're in an independent research project, we like discuss what we could do, and then discuss ideas, and then we would pick whatever we were interested in. And when I was really interested in something, I actually put lots of effort and time into it instead of something that I don't really like and just doing it just for the sake of doing it.
For me, I found out about YCLI through my AP Environmental Science class. My teacher brought it to our attention as a class and offered us like extra credit if we applied. But I mean, I did it because it looked fun. So I guess just like having teachers recommend it is a nice way to get involved.
Can the old man offer a suggestion?
For sure.
I have one word, and that is read. Read everything. Read books. Read newspapers. Read the ingredients in a soup can. Read everything. Because the more you read, for one thing, the better your vocabulary becomes, but also the more you learn in a wider range of topics. And it's important, I think, to learn not only your science, but also to learn the literature that our language has. And the more you do that, the better you are at explaining what it is you want to say. You all did a wonderful job, and I'm impressed with your skills. But I think that if you read more, it would sound better. Just a comment.
I think they did a fantastic job considering this is not a dissertation. This is a school project and something that I think it demonstrates the curiosity, which is something that all scientists need to have. They need to have the question of why. Why does something happen? What is happening there? What is going on? I think reading is absolutely a fundamental part of everything that we do, for sure. And that's what helps. But to have that initial curiosity to want to go and understand more is certainly a big component of it as well. So thank you all very much again. And Harry, thanks for your comments. I have one other question, and I've asked this before, and it has helped honestly inspire me in a lot of the work that we do and the kinds of things that we try to do as part of the district. But I want to ask Becca and Cynthia and Nimesh and Carolyn, why do you do this? What helps to inspire your curiosity? Carolyn kind of mentioned it a little bit ago, and it caught my ear, made my ear perk up. But why do you do this when you have so many other things that you could be doing? Why do you choose conservation and environmental as a way to work and projects and things like that? Why do you choose that to spend your time? Can you talk a little bit about that?
I can go first, Lisa. So honestly, for me, I got really into marine science when I was younger. Because I used to live in like Japan, specifically Okinawa, and I was surrounded by like water and like reefs. And I loved going to the aquarium and just like learning new things about it. It was such an interesting topic to me, and I wanted to learn more about it. And by learning more about it, I started like realizing like how much danger like the ocean's in. And so like I just have like in me like I just want to like help. Sometimes a little too passionate. I kind of overthink things. I kind of like, as Ms. Palmer knows, I came in with a very, very broad topic and a lot to narrow down, but I don't know. I've always just really liked this topic. It's something that I'm very, very passionate about. And I think I'm going to keep researching and keep looking into it throughout my life. Just kind of stemmed from when I was younger.
Excellent. Excellent.
I guess for me, like what Harry said about, reading, I did a lot of, well, at first, like when I wasn't really aware about this issue, I did a lot of reading and researching about this. And at that time, like it was pretty new to me, but after doing a lot of research, I really understood that, like this is something I really want to change and take part in because it's such a pressing issue today. And I just thought that, like I have to do something about it. So, and then the more I got involved with it, the more interested I got. And this year I'm also taking AP Environmental Science, so that's like definitely one of my favorite classes because I'm really able to do it not only outside of school, but in school as well.
For me, it was environmental science is a way that I can contribute to the community and actually have an impact. Because when I actually take on an environmental science project, I feel like I'm actually doing something that can really help the environment, help people's lives, and that can actually positively contribute. So I just enjoy the fact that I'm able to do it.
I've always been super passionate about the environment. I was first exposed to by my grandfather, who was also super passionate. And then in 7th grade, I had an amazing life sciences teacher. And when we learned about conservation and environmental science, I just thought it was just so interesting. Like it was just so cool and so relevant and always so important. And the more I got involved, the more I realized how interested I was in it and how much of a difference even small things can make in bettering our entire world. And so the more I got involved, the more I wanted to keep getting involved.
That's excellent. Well, thank you all so much. As I mentioned, I think many years ago, we did a program on young environmental leaders, and it included representatives from Envirothon teams and the student representative to EQUAC. And I asked that same question: what inspires you? Why do you do this? Why, when there's, all sorts of other things that could be occupying your time, why do you come and work on environmental issues? And the primary reason at that time was it was fun. And what I'm getting goosebumps just thinking about it is it's fun and you're making a difference. And that's exactly what we all are hoping, to do. And that's why I think all 51 of us that are sitting here on this call today, that's exactly the kind of inspiration we're looking to take away, is we can make a difference. So again, kudos to all of you. You get an A+ in my book in regards to all of the different methods, the projects, the thoughtfulness, and everything that went into these projects. You may feel like we've got a lot of pressure putting on you. You don't. You've got a lot of us that are here to help you and to help continue to inspire you in making a difference in our environment. And you can make a difference for sure. And we look forward to continuing to work together. That's what will make the difference, is our engaged community that is working together. So thank you all very, very much. I'm going to give it back to Ashley if you have any parting thoughts or any final questions.
I don't have any last questions. I think these guys have been questioned out enough for Saturday morning. I'll just thank you again, Becca and Cynthia and Nimesh and Carolyn and all of the people who have joined us for this webinar today to see what they're working on, because I think that your work has been incredible. If you look in the chat, everyone says that you're inspiring, that this is excellent work, that they're so proud that this kind of thing is coming out of Fairfax County, and we are too. It's been a pleasure to work with you. Some of you I know have applied for YCLI this year, so I will hopefully get to work with you again this year. And should you ever want to do another project like this, the district will always be here to support your research interests and things that you're passionate about. And so we're so glad that we got to invite all of you guys today, and hopefully we'll get to work with you again in the future.
Thank you very much, Ashley. Again, I get the good fortune of being able to extend kudos to you, and thank you again for all of the mentorship that you provided to these wonderful students, those that are here today, and those that we also reach out to throughout the course of the year. I wanted to just wrap up by saying we are going to be having our November Green Breakfast on November 14th. Stay tuned for a little bit of information about that. We're hoping Michael Bishop does come and join us to talk a little bit more about the Northern Virginia Purple Martin initiative as well. And we look forward to seeing you then. Again, because of the pandemic, we are going to be virtual for the time being. We do certainly miss seeing everybody in person and enjoying that wonderful breakfast over at Brian's Grill. I realize I should put my video on here so you can see me too. It's nothing like talking to a strange space here. But we do miss you, and we do look forward to again seeing you all soon once this is all behind us and we're able to gather in large groups again. But again, thank you all very much. We look forward to seeing you virtually on November 14th. And until then, be safe. Enjoy the beautiful fall weather and the changing of the leaves, and we'll see you soon.